Thanks everyone for coming. I'm going to present some work that we published last year in PLOS Biology on doing sequence analysis to understand combinatorial regulation in Drosophila development. But before I go to the main part, the main topic, I'd like to begin with a specific bioinformatics task that Magnus was also talking about. That's going to be key in the rest of the talk, and the task is to predict transcription factor binding loci in the genome. So the first step in doing this is to determine the binding preference of the transcription factor. For example, by collecting examples of binding sites and then summarizing their pattern in some sort of a probability or frequency matrix, which is often called a position weight matrix or PWM, also generically called a motif. And I'd like to share with you a wonderful resource from my collaborators at UMass Med for motifs in Drosophila. This is Fly Factor Survey, which has experimentally characterized motifs for 326 transcription factors in Drosophila and is still growing. So once you have the binding preference or specificity of the transcription factor, you can scan the genome for matches to the motif using standard methods available. You can look for exact matches to the pattern or approximate matches. These standard methods are known to lead to several false positives, and so one of the ideas that has been around for nearly 10 years now is to look for statistically significant clusters of binding sites. And this idea can be formalized or formally implemented in a hidden Markov model, which is what we did in this case. And with that approach, you can scan the genome in windows of a fixed length and score each window for the presence of one or more weak or strong sites using the hidden Markov model. And you'll get a genome-wide score profile that looks like this for your motif. And by the way, you can do this for each of the 326 transcription factors at Fly Factor Survey through a separate interface called Drosophila Genome Surveyor that we've made available. And so the next idea that we use to improve the accuracy of predicting transcription factor targets is that of comparative genomics. And the idea, again, is very intuitive. You scan your motif not only in Drosophila melanogaster, but also in several other closely related species. And then you try to combine the scores from orthologous windows in those species into one score for your motif. And hopefully you'll end up with a much cleaner profile that's based on comparative genomics and that reflects the extent of functional binding of the transcription factor at any locus in the genome. So the question is, how do you combine these multi-species tracks of the same motif into one track, which is shown here at the bottom? You could compute a simple average of those scores from orthologous windows. And of course, this is very easy to do, but it also has the problem that it ignores the phylogeny completely. And if you have an uneven phylogeny, that could lead to problems. And so the other option is to somehow come up with a phylogenetically weighted average of the scores of orthologous windows. And so that's what we did here. The method we used is described in this slide. Let's assume that we have 4 species and that are related by this phylogeny, and the HMM scores for a motif are X1, X2, X3, and X4. These are the scores at the extant species, and these are the scores that we want to combine into one score. We imagine that xt is a random variable evolving along the branches of this phylogeny as per a Brownian motion process, so that the value of the variable at any node has a Gaussian distribution conditioned on the value of the variable at its parent node. You can define the evolutionary or temporal average of this variable over the entire tree. It's simply an integral of the variable over all the branches divided by the total time span. And then you can compute the expectation. So this is a random variable in itself, because xt was a random process. You can compute the expectation of this temporal average given the observations x1, x2, x3, x4 at the extant species. And so this is the final phylogenetic average of the scores that we use to predict the amount of motif presence in any given window in the genome. And by the way, this idea is not new. It was used in bioinformatics by Stone and Saido in 2007, and our contribution was to come up with a faster implementation using the upward-downward algorithm. So once we have a score like this that combines multi-species information and gives you one profile for a motif's presence in the genome, you can, the first thing to do is to see if it's working well. So the way we validated this, and much like Magnus was talking about, is to compare to chip data. Ideally, the computationally predicted scores should match the chip intensities. And so there are many ways to do that comparison. We performed a statistical test where we took chip data, we took the top binding regions from the genome and non-binding regions from the genome and compared the motif-based scores between those regions, and thus got a p-value for each chip track. So there were 8 transcription factors for which we had data at that time, and these are the p-values from a Wilcoxon test showing that in all cases the discriminative ability of the motif-based score is statistically significant, and it's clearly much more significant in some cases, such as Bicoid and Huckabeen, compared to some others, such as Hunchback and Knirps. But importantly, if you do the exact same thing with motif scores coming from 2 species comparisons, you see a clear improvement in the discriminative ability, in the agreement between chip data and motif-based scores. And when you do the same using the phylogenetic weighted averaging from 11 Drosophila species, you see a very spectacular improvement in that discriminative ability. So this is where I'll stop with this part of the talk, which is how to predict transcription factor binding loci in the genome. To recap, we are using an HMM to score for binding site clusters, and we are combining the scores from orthologous windows in 11 different species to get one score for each window. And that's the score we use in the rest of the talk. So now we are ready to move on to the main topic of the talk, which is to understand enhancers or CRMs and to quantitatively model their function. And I'll explain what I mean by that. So a couple of slides on introducing you to cis-regulatory modules, also called enhancers in Drosophila. This is the classic example from Drosophila development, the even skip gene with its 7 stripes of expression in the blastoderm stage embryo. So I'd like to point out that I'm talking about spatial expression patterns. This is, we have the embryo here. These are the different, they're composed of many nuclei, and the lighted up cells, the lighted up nuclei are those where the gene is expressed. So this is spatial expression patterns as opposed to temporal expression patterns that you saw in the previous talk. Now, a lot of painstaking work in the wet lab has shown that each of these different stripes of expression is regulated or caused by a separate cis-regulatory sequence. For example, there's a special sequence for the second of those stripes, famously the famous Eve stripe 2 enhancer. And so here's a genome browser view of the same gene and the Eve stripe 2 cis-regulatory module. It's about a kb in length, about a kb away from the gene, And if you look into it, you'll find many binding sites. You'll find binding sites for activators, bicoid and hunchback. You'll find binding sites for repressors, cruple and giant. And you'll find many binding sites for the same transcription factor as well. So homotypic as well as heterotypic clustering of binding sites. All of these acting together to drive that single E-stripe 2 pattern that you saw. And these are called cis-regulatory modules. Why modules? Because if you cut this piece out and attach it to a reporter gene, it'll recapitulate the stripe 2 expression. And so the information about this expression pattern, the fact that the gene will be expressed at this interval along the axis, is included in the binding sites within this piece of sequence. That's why these are called modules. I'll be calling them CRMs. So the task that we are addressing in this paper is first to model CRM function. That is, given the sequence of a CRM, can we predict that it'll drive this expression pattern? And for this, we have to get into the details of combinatorial regulation that's going on within that CRM. But that's not all. That's not the only piece of information we need. We need more information than that. And let me explain it to you in the context of the regulatory network that we studied, which is the anterior-posterior patterning network. This is the series of regulatory processes that result in segmentation of the Drosophila embryo. And the regulatory network consists of several transcription factors. The x-axis here is the AP axis. So this is the left end would be the head, the right end would be the tail, or the anterior and the posterior. And there are some transcription factors that are expressed in the form of a gradient along the axis, and some transcription factors are expressed at specific intervals or gaps along the axis. And all of these factors act together in various ways to drive the enhancers or CRMs. And those CRMs in turn drive the expression pattern, the spatial expression pattern of the nearby genes. And so what's not understood very well is how exactly that combinatorial regulation is happening. And what we'd like to do is given all this information about the transcription factor concentration profiles and the information of the sequence of a particular CRM, we'd like to predict the function. So this is the problem at hand. And in fact, we'll simplify the problem by boiling it down to a one-dimensional expression pattern. That is, instead of looking at every nucleus, we are the E-stripe 2 pattern will be simplified as being expression at this interval along the axis, and the goal would be to predict this kind of the correct location of the expression. And what we did was a very simple approach. The hypothesis was our approach to modeling the function of a CRM is this. We hypothesized that to a first approximation, the output expression pattern of a CRM is a simple function of the counts of activator and repressor binding sites and their respective concentration levels. The model is going to be simple. In particular, it's going to be a generalized linear model, and simple. One way to look at it is that there are fewer parameters involved as opposed to some other models that have been published recently, which use statistical thermodynamics, and we've been working on such models as well. But what I'm going to talk about here is this simple logistic regression-based model. So I'll spend some time on explaining how the model works. First of all, we use the first part of the talk to predict motif presence at each locus in the genome. And let's say this is the CRM. Here's the gene and here's the CRM whose function we want to predict. We take each of the motifs given to us and measure their presence in that CRM using the method I described earlier, the HMM-based method using comparative genomics. Once we have a motif count, we then multiply the motif counts with the factor concentration profiles. So these are the transcription factor concentration profiles which I mentioned earlier. Some of them are in the form of gradients. Some of them are sharp regions of expression. So we multiply the motif counts with the transcription factor concentration profiles and a certain weight, one weight parameter for each transcription factor that reflects whether it's an activator or repressor. Activators get a positive weight, repressors get a negative weight. And get what we call a weighted occupancy profile for each transcription factor. And then we combine these weighted occupancies in a simple, through a simple logit function and get the predicted expression profile shown here in dark blue. In this case, it matches pretty well with the real expression profile driven by the CRM, which is these two red bricks here. So that's the model, and in particular, while training the model, we'll learn one parameter for each transcription factor that'll tell us whether it's an activator or a repressor. We trained the model on 46 CRMs for which we had sequence and expression data, and the model, the best fit model, gave fairly good predictions on 20 of those CRMs. For example, here, by the way, the red is the real expression along the axis, the dark blue is the predicted expression. So in 20 of the CRMs, the expression matched very well. On about 15 of them, the predicted expression had a fair match, so there were some aspects that we were missing. For example, here we are missing the dip in the expression between the stripes 4 and 6 of even skipped. And there were 11 of these CRMs for which we were completely off. And I'm not going into more quantitative assessment of the model, using statistical methods, just to give a feel of how well the model works. But once we have this model trained, we can do fun things with it, which biologists care about a lot. And in particular, I'll show you how to use it for predicting novel CRMs and predicting interactions between transcription factors and CRMs. So the first task I mentioned was CRM discovery, and I'll spend a couple of slides on the traditional approach to this. To predict a transcription factor target, you take the motif and you scan the genome. I've shown this earlier. You can do the same for each of several different motifs. And CRM discovery tries to combine this information into one track, one score profile somehow. And there are simple methods such as averaging over motifs or advanced methods that are based on multi-motif HMMs that you can use for this purpose. But this is an entirely sequence-driven approach, and it's not clear what are the optimal combination of motifs that we should be using. Why these 4 motifs in order to predict CRMs? Similarly, in order to predict interactions between transcription factors and CRMs, what you could do is go to a CRM, note down which motif has a strong presence in that CRM, and based on that, you say that the corresponding transcription factor is regulating that CRM or the gene. But again, what if that transcription factor is not expressed where the gene is expressed? So the lesson from both of these examples is that we need to consider transcription factor concentration or expression when predicting CRMs or when predicting individual interactions between transcription factors and CRMs. So we devised a new method to predict CRMs using sequence as well as expression data. We called it pattern generating potentials, and the way it works is this. Let's say we have a gene here, the SLOPy2 gene, and this is the intergenic locus, 20 kb around the gene. We know the gene expression profile. We know that it is expressed at this position along the anterior-posterior axis of the embryo. We scan a window of a kb length along the locus, and for each position of the window, we use our quantitative model to predict what expression pattern it should drive. And let's say this window drives this pattern, which looks a lot like this one, the endogenous gene expression pattern. And so we give this window a high score. That's the score profile, the pattern generating potential score for this particular window, because it's predicting an expression that looks like the gene's expression. And we keep doing this. This window, for example, when we predict its expression profile, doesn't match the endogenous profile, and so we give it a low score, and so on. It took a little bit of work, a fair bit of work, to come up with the right comparison function itself, how to compare two expression profiles. But when we were satisfied with what we had, we implemented the final scoring method and had a CRM discovery approach that uses transcription factor expression information or concentration information. And we compared it to sequence methods based on sequence alone, and I won't explain what exactly this is, something like an RSC. And it outperformed all these sequence-based methods, both ours and external methods. When we ran the method to discover new CRMs, we found an interesting phenomenon. First of all, so what this shows is a particular gene and its expression pattern. So this gene has expression here, and each row below it would be a separate predicted CRM and its expression pattern. So, for example, for this gene caudal, we predicted 3 CRMs. One of them is a known CRM that drives expression right about where the gene is expressed. But there are 2 more that also do the same thing. And the same theme of multiple CRMs driving the same expression pattern was found in a significant number of genes. So for example, for nubbin here, there were 4 different CRMs predicted that should be driving the same expression pattern. One of them is known, the others are not known yet. And so this theme of redundant CRMs was interesting because of the recent reports of shadow enhancers in the Drosophila community, exactly this phenomenon. So then we went ahead and extended this basic idea to apply to large numbers of genes, and for this we exploited a database of in situ expression images. The Fly Express database maintained and developed by Sudhir Kumar, our collaborator. So what we do here is, this database has in situ expression patterns for about 6,000 or 7,000 genes, and you can query a gene, in this case AMA or amalgam, and you'll get many expression patterns at the blastoderm stage. In this case, you summarize all that information automatically to get this kind of a one-dimensional expression pattern for the gene. And then you run PGP, our method, in the intergenic locus around that gene, and you get predicted CRMs for that gene. And so doing that, we predicted a large number of CRMs, new CRMs, for anterior-posterior pattern genes from the Fly Express database. Again, this is showing the known expression pattern and the predicted expression pattern of a predicted CRM. Our collaborators, Mike Brodsky and Sue Selniker, helped validate experimentally a handful of these CRM predictions, and the handful that we tested worked as expected. But beyond CRM discovery, we also went ahead and tried to predict individual interactions between transcription factors and genes or CRMs by doing in silico knockdowns, if you will. So I'll illustrate that with an example. Here's a particular CRM, KNERPS KD. This red bar is the expression pattern of the CRM along the AP axis, and the blue is the prediction from the model. So it matches reasonably well where the CRM drives expression. We then can repeat the model's predictions after knocking down one of the transcription factors, and you get this. This suggests that this particular transcription factor is an activator, because if you knock it down, the predicted expression goes down. Likewise, if we knock down a different transcription factor, hunchback, we get extensive de-repression from our predictions. And so you would predict that this is a repressor for the CRM. And so doing this and doing some permutation tests to assess statistical significance of these effects, we got these kinds of umbrella diagrams explaining how this pattern of expression is achieved. So caudal is an activator, hunchback is a repressor in the anterior end, and so on for this CRM. And in turn, and you'll get an entire gene regulatory network where edges represent interactions between individual transcription factors and the CRMs. A large number of these interactions were already validated from the literature, but there was an equally large number that are novel predictions. For example, one piece of this network is driven by this transcription factor called Capicua. So this is the predicted regulatory network of direct and indirect targets of Capicua. This is a transcription factor that was not well known to be a major player in the AP patterning network, and in fact, only last year, just after our paper, a couple of papers have come out experimentally showing some of these edges to be true. And to end, so I'm done with what I wanted to show. To end, there's a short note on using chip data instead of motif-based scores for all of these tasks. So you can repeat much of these tasks by replacing the motif scores with chip data for the same transcription factors. We did that, and the results were surprising. So the chip data was worse, actually significantly worse in predicting expression patterns. We had a way to assess how much of the occupancy, predicted occupancy, is functional, and it's a statistical method for assessing this. And even there, the motif-based predictions seem to be better, partly because they're using comparative genomics information. Chip data is not. For a couple of transcription factors, Knurps and Giant, the chip data gave really unreasonable-looking results. And predicting TF-CRM interactions, you would normally think that if a chip peak is present, that's a regulatory interaction there. The results were actually, again, surprising. They were worse than that based on our model and sequence analysis. I'd like to thank Majid Kazemian and Charles Platty, the grad students who did this work, my collaborators Mike Brodsky and Scott Wolf, who did the fly experiments, Sudhir Kumar, who maintains the Fly Express database, and Sue Selnick at Berkeley, funding from NIH. Thank you.